everyone, I'm Tessa O'Connor, and the following is a presentation on a review paper that summarizes the recent advances in the research of environmental factors that influence the risk of autism spectrum disorder. This paper was published in 2017 in the Journal of Research in Medical Sciences. Although I discussed the ASD phenotype extensively in my previous presentation, I wanted to include a quick summary here. This disorder is characterized by deficiencies in social and behavioral communications, language impairment, and restricted repetitive patterns of behavior. The authors of this paper point out in their introduction that the lack of complete concordance between monozygotic twins indicates that environmental risk factors play an important role in the etiology of ASD. The authors also indicate that the major mechanism of ASD etiology is then suggested to be interactions between genes carrying risk for ASD, as well as environmental factors. Epigenetics is suggested as kind of the bridge that connects the environmental factors of ASD to the genetic factors. The authors highlight that environmental factors can change the expression of genes through epigenetic mechanisms such as DNA methylation, histone protein changes, and the expression of non-coding RNAs. These epigenetic changes can be passed on through generations of cells or even organisms, which is how environmental factors can lead to changes in the expression of genes with ASD risk in developing embryos. The authors divided the environmental risk factors of ASD as prenatal, natal, and postnatal factors. The prenatal risk factors of ASD, as identified by the authors of this paper, are parental age, maternal physical and mental health, maternal prenatal medication, and familial socioeconomic status. The authors cited numerous studies that implicate parental age in the risk of ASD development. Many of these studies have found that parental age over 34 years old is associated with increased ASD risk. Most of the studies attribute more risk to increased paternal age rather than maternal age. One such study indicates that there's a 29% increase in risk for ASD with every 10-year increase in paternal age. The ASD risk associated with increased paternal age could possibly be attributed to the increased occurrence of de novo mutations in germline cells, as well as the increased modifications of DNA via methylation. These could potentially result in the altered expression of genes involved in neural development, as well as disorders in sperm genomic imprinting. These would both increase the risk of developing a neural impairment such as ASD. Studies that have identified advanced maternal age as a risk factor in the development of ASD have highlighted three predominant causes: chromosomal abnormalities, trinucleotide expansions in ovules, and increased obstetric intervention. Trinucleotide expansions refer to duplications of a trinucleotide sequence that occurs in ovules, an example of which can be seen in the diagram here. Studies summarized by the authors of this paper also identified multiple physical health conditions that increase the risk of developing ASD. Notably, metabolic syndrome, which can include diabetes, high blood pressure, and/or obesity, has been identified as significant. Further, maternal bleeding during pregnancy was determined to increase the risk of autism by 81%. Both of these physical conditions can lead to hypoxia in utero, which means there's a lack of oxygen for the fetus. This can inhibit brain development by impacting myelination of neurons, membrane adhesion, and the number of hippocampal neurons. Viral and bacterial infections during pregnancy can also increase the risk for ASD because they result in abnormal maternal immune system activation. This activation induces elevated levels of inflammatory cytokines, which affect the brain development of the fetus and increase risk for ASD. Additionally, studies have identified factors that relate to maternal mental health that carry ASD risk. Environmental factors such as parental behaviors and communication patterns can impact the development of a child's personality and emotions. A history of mental illnesses such as schizophrenia, depression, anxiety, and personality disorders in the parental unit can also increase the risk of ASD. Significant stress during the 21st to 32nd weeks of gestation has also been identified as an ASD risk factor. Specifically, this stress can induce epigenetic modifications that change the expression of genes that neurobiologically, metabolically, and physiologically respond to stress in the fetus. The authors of this paper also indicated that prenatal medication use by mothers has been linked to ASD development. In particular, psychiatric medication use increases ASD risk by 68%. These medications can cross the placenta and disturb fetal development. Antiepileptic drugs have been identified as particularly harmful. These can increase oxidative stress in the fetus and lead to varied gene expression patterns. These effects can result in developmental delays and deficiencies in motor movements and social behaviors. Interestingly, the authors presented convergent evidence that socioeconomic status is an ASD risk factor. Families of low socioeconomic status experience unhealthy sociality and unrehabilitated life conditions, as well as less access to health care. The increased psychological stress experienced in low-income families can also increase the susceptibility of a fetus or a child to developing ASD. This table summarizes the prenatal risk factors reviewed by the authors and the studies that support the negative and/or positive correlation of each factor to ASD. Of note, many studies agree that advanced parental age, paternal age, and maternal age, as well as maternal bleeding during pregnancy, anxiety and stress, antiepileptic drugs, and social status all have a positive association with ASD development. The natal factors associated with ASD development are far fewer than the prenatal factors. Abnormal gestation age, both preterm and postterm, has been found to increase ASD development. Birth complications that include fetal distress, umbilical cord complications like fetal nuchal cord, where the umbilical cord wraps around the neck of the fetus, and C-sections all have the potential to cause hypoxia. Hypoxia can lead to damage in the nervous system, increasing ASD risk. Hypoxia is also known to cause large lateral ventricles, morphological hippocampal abnormalities, and increased dopaminergic activity, which have all been identified in the brains of children with ASD. This suggests a correlation between hypoxia and ASD brain development. This table illustrates the natal risk factors associated with ASD as determined by the authors of this paper. Of note, preterm births, C-section deliveries, fetal complications, hypoxia, and umbilical cord complications have strong convergent evidence of positive associations to ASD development. The postnatal risk factors of ASD, as identified by the authors, are also far fewer than the prenatal factors. Low birth weight, being defined by a weight of less than 5.5 pounds, has a twofold increase in risk for ASD. Postnatal jaundice is a sometimes lethal condition for a newborn and can lead to a fourfold increase in risk for ASD if it is survived. Jaundice results from a high bilirubin accumulation that is caused by an increased breakdown of red blood cells and a low excretory capacity of the liver. This is usually caused by an immature liver in newborns. Finally, postnatal infections such as meningitis, mumps, and chickenpox within the first 30 days of birth have also been associated with ASD development. This table lists postnatal risk factors and their association with ASD as identified by the authors. The independent studies that identified a positive association of low birth weight with ASD are numerous. However, many studies identified a negative association too. Jaundice and postnatal infections, as well as birth defects, also have convergent evidence of a positive association with ASD. Finally, the authors identified some factors that do not directly impact risk for ASD, but rather play protective roles in ASD development. Unsaturated fatty acids, specifically linoleic acid and omegas 3 and 4, are significant protective factors during pregnancy for ASD development. The effects of these fatty acids include contributing to retinal and brain development in utero, signal transduction, gene expression, and they are cell membrane components. These fatty acids are particularly important during the first 2 months of pregnancy when the most critical physical development is seen. High intake of these fatty acids during pregnancy has been determined to reduce the risk of ASD by 34%. Studies have also determined that folic acid supplements can help protect against ASD development. Folic acid is used to synthesize DNA, and it's also needed to help the maturation of red and white blood cells. Folic acid intake during pregnancy has been associated with less behavioral problems in infants, reduced risk of language deficiencies, improved verbal and attention competencies, and a decreased risk of ASD. Here's the reference for the paper, as well as a reference for one of the diagrams I used. Thanks for watching!